Infectious disease, something so common in our everyday lives, yet not understood by humans until relatively recent history. Infectious disease was just a given fact of life, a curse, an unfortunate turn of fate, or a supernatural force that decided who lived and who died. Human health was still mainly thought to be determined by humors within the body that were tied to celestial bodies, seasons, and stages of life. Many believed an unbalance in these humors was what caused diseases and illnesses. The existence of microbes was discovered in the mid-1600s, but not many had theorized that they may have had something to do with infectious disease, and certainly nobody had proven this. This all changed when a French tanner's son developed an interest in microbes. His name was Louis Pasteur. In the 1800s, Louis Pasteur proved that microbes are responsible for infectious diseases. He demonstrated how important microbes' roles are in everyday life and how the creation of a vaccine may prevent or lessen the effects of an infectious disease. He pushed the frontier of microbiology, and many consider him to be the father of modern medicine. At the time of the early 1800s, the common knowledge of how microbes appeared was a theory known as spontaneous generation. A concept thought up by the great Greek philosopher Aristotle. Spontaneous generation was the idea that microbes and even small pests or plants materialize out of decaying matter. At this point in history, the idea that rats and maggots were generated out of decaying matter had already been disproven, but the theory still stood for microbes. A Dutch tradesman by the name of Antonie van Leeuwenhoek made the first discovery of microbes through a microscope. Microscopes were the key to seeing such tiny organisms, and the progress made in microscope development had taken great leaps within the last 2 centuries. In 1765, Lazzaro Spallanzani discovered mitosis. In 1796, Edward Jenner had happened upon the discovery that workers exposed to cowpox had minimal symptoms when then infected with smallpox, a deadlier disease. He then used injections of cowpox to help reduce fatality rates of smallpox, and this was the very first vaccine. Louis Pasteur was born on December 27, 1822. His father, Jean-Joseph Pasteur, is a veteran that fought for Napoleon and has settled into the role of a tanner. Contrary to Pasteur's genius in later life, the boy Louis struggled in school and showed no signs of elevated intellect. He went to the Royal College of Besançon and graduated with a Bachelor of Arts and a Bachelor of Science. He then received a doctorate from the École Normale in 1847. Pasteur spent several years teaching, and in 1849 he begins research on molecular asymmetry in crystals, namely tartaric acid. He made an important discovery in the difference between tartaric acid and paratartaric acid. This was Pasteur's first significant contribution to science. While working as a professor of chemistry at the École Normale Supérieure, Pasteur was approached by one of his pupil's fathers. He asked Pasteur for help with the recent sugar beet alcohol problem. Batches of sugar beet alcohol were either going bad or fermenting with a sour taste. Pasteur began his work on alcohol fermentation in 1856. And he took samples of good batches and bad batches of the alcohol. He discovers that brewer's yeast was a microbe. Before this, many thought yeast was a plant or fungi. He also found other microbes in the batches that produced a sour taste. He isolated this microbe and concluded that this microbe was harmful to the yeast. Pasteur went on to discover that when the fermenting substance was heated to 50 to 60 degrees Celsius for a short amount of time and alcohol was passed through, the harmful microbes were killed and the alcohol would ferment successfully. The process of pasteurization saved many breweries and distilleries because they now knew what was causing their alcohol to go bad. Pasteurization is an important process that is still used today. In 1858, Pasteur begins his research on spontaneous generation. He disagreed with the theory, bringing him into the ongoing debate. The idea that the common knowledge of spontaneous generation was false was a threat to the established order, and some scientists like Félix Pouchet wrote papers proving it. Pasteur devised an experiment to disprove this prevailing theory. He took swan or gooseneck flasks and filled them with nutrient solutions. Pasteur then heated these flasks to purify them of any microbes. After purifying, he passed unfiltered air into the opening of the flasks. If spontaneous generation was true, the microbes would appear in the nutrient solution. After taking samples of the solution and the dip in the gooseneck, Pasteur found no microbes in the nutrient solution, yet found many within the dip. He published his findings in 1862 and was awarded the Al-Humbert Prize for his work. In 1865, Pasteur patented his pasteurization process. He then goes on to investigate a disease that had been ravaging the silkworm industry in France. He discovered some silkworms with globular growths and concluded that only the silkworms without globules should reproduce. This did not work, and Pasteur faced harsh criticism for the next 2 years. He eventually discovered a second disease that was killing the silkworms, and his work laid the foundation for epidemiology. In 1877, Pasteur begins research on anthrax, and by 1881 he had developed a vaccine for it. In May of 1881, Pasteur staged a bold public demonstration of his new anthrax vaccine. He gathered 50 sheep in a field at Puy-les-Fort and split them into 2 groups of 25. One group was injected with his vaccine, and while the other was not. He then injected the blood of sheep dead from anthrax into both groups and said that within 48 hours, the 25 sheep not vaccinated would be dead. And sure enough, at the end of those 48 hours, 23 unvaccinated sheep were dead, and 2 were weakly bleeding on the ground, dying. The other 25 vaccinated sheep were still alive and healthy. By the end, a crowd of more than 200 had gathered. Not 2 weeks after a successful test, Pasteur published his finding to the Académie des Sciences. This vaccine was made with knowledge from Pasteur's previous vaccine, one used to prevent chicken cholera. Both were made through a process called attenuation. An attenuation of a microbial culture is when the virulence of the culture is reduced. This can be done by killing off some of the microbes or passing through another organism, and tissue sample is taken from the infected organism. In Pasteur's case, he attenuated his cultures of anthrax and chicken cholera through atmospheric oxygen attenuation, which killed off a portion of the microbes in the culture, reducing its force. By 1885, Pasteur was working on yet another vaccine, this time one being developed for use in humans, the rabies vaccine. Pasteur created this vaccine using the process of attenuation again, but this time through rabbits. He would inject rabbits with a virulent strain of rabies, and when the rabbits died, he would grow a culture of the now attenuated rabies with their spinal tissue. He had conducted multiple tests on rabid dogs with successful results. On July 6, 1885, a 9-year-old boy by the name of Joseph Meister and his distressed mother arrived at Pasteur's lab in Paris. Meister had been badly bitten by a rabid dog a dozen or more times, and the worst of his wounds had already been cauterized by a local smith. After analysis of the boy's wounds and consultation with colleagues, Pasteur concluded that the boy would surely die from rabies without intervention, and this is when Pasteur decided to administer his new vaccine. This was the very first human rabies vaccine, with 13 injections in 11 days. The boy survived the disease he once was thought to be doomed from. At the beginning, the idea of little invisible creatures was preposterous to many, and some called Pasteur mad, or some scientists like Justus von Liebig and Jacob Berzelius even ridiculed his work. But during this time, Pasteur became the modern-day equivalent of a rock star, a household name and a perfect example of a scientist. His anthrax vaccine was in high demand by many soon after its invention. Farmers all over wanted to get their livestock vaccinated. Similarly, the creation of the rabies vaccine was also in high demand shortly after the first successful tests, and this brought in a large profit for the newly founded Institut Pasteur. Some people, though, criticized Pasteur's work. Many claims of people dying after being administered the rabies vaccine. Many of these claims were refuted and ultimately drowned out by the pro-Pasteurians. Some of the consequences of Pasteur's work were immediate. Pasteurization was used by many brewers soon after its discovery, but even to this day it is still used in many everyday products. Not only is it still used in alcohol fermentation, it is also used in pasteurizing most dairy products such as milk, cheese, and yogurt, and is also used in eggs, juices, and flour. His anthrax vaccine was highly sought after by farmers all over Europe that wanted to vaccinate their livestock. The demand for his rabies vaccine also rose sharply after the first successful human trials. It was not without controversy either. Many thought the vaccine was dangerous. Even though modern-day vaccines are not similar to Pasteur's dead vaccines instead of Pasteur's live ones, many concepts are the same, including the controversy part. Though ironically, a new rabies vaccine is in development that is in fact a live vaccine, just like Pasteur's. By the time of his death in 1895, Louis Pasteur taught us that microbes cause disease, microbes have a very significant role in everyday life, and that a vaccine can be created to prevent a disease. He was a tireless worker that worked through criticism and controversy, pushing the frontier of microbiology and laying the foundations of modern hygiene and medicine.